glad to introduce today's speaker, Professor Huang Yixue. So, Professor Huang, she received her PhD degree from University of Texas, Southwestern Medical Center in Dallas. Later, she completed postdoc training at Dr. Joe Richard's lab in UMass Medical School. She's now a research fellow and also serves as a deputy director in an Institute of Medical Science in Academia Sinica, 中央研究院的生物医学研究所的副所长。Okay. Dr. Huang specialized in molecular neuroscience and with a particular focus on how medical translation can regulate the learning, memory, and other physiological status. Her lab work on protein, which you will learn a lot later. So cytoplasmic polyadenylation element binding protein, CPEB. Okay. This protein they do a very sophisticated translation to promote the neuronal plasticity. Dr. Huang's work have appeared in many prestigious journals, and she contributed to our understanding of the neuron plasticity. So in addition to her academic achievement, she also served as to our scientific community. She's the senior editor of the journal Brain Research, and she will soon become the president of the Taiwan Neuroscience Alliance. So her topic today is translational control in memory and metabolism. Please join me to welcome Professor Huang.
Dr. Song, for such a kind introduction. I'm glad to be here to share the work in my lab. And I'm so impressed to see so many students here. Because a lot of times we only teach few students and teach a lot. So I hope my talk is clear to the students. So the first part of the talk, I'm going to talk about the lab. Initially, what we want to work on is to study how the long-term memory is formed and what kind of molecules are required for that. But back to that, actually, I'm coming from the RNA biology lab. We actually study molecules. It's just to put this molecule in the context of the neuron and the physiology of the memory. So just a quick reminder for messenger RNA, as you all know, they contain the open reading frame, and this has a message important to encode the protein for physiological function. On top of this, you have 3' poly A tail and 5' M7G cap. Both of the structure are very important to keep the RNA stable inside the cells to prevent the degradation from S-ribonucleus. And in addition to this, we also have a 5' and 3' untranslated region. So the M7G cap is important is because the initiation factor 4E has to recognize this structure to assemble a lot of translation initiation factor. And also the 3' poly A tail can be bound by poly A binding protein. This also helps to recruit the EIF4G. That's why when we see the elongate poly A tail and also the M7G structure is also important for translation, not just important for RNA stability. So for us, we only care about how RNA is translated into the protein. Can this process be regulated? So most of the time we focus on the 3' and 5' untranslated region because that's where all this regulatory information is there. So especially for the protein I work on, cytoplasmic polyadenylation element binding protein, For its name, it's because they bind to a sequence we know is called the cytoplasmic phosphorylation element, and initially was identified from the Xenopus oocyte. That's the lab where I was from. So during the oocyte maturation, there's a lot of gene regulation. It's dependent on translational control. From there, we identify this element, and we identify the initial binding protein is called the CPB1. And because later on there's a family protein appears, so I kind of focus on this 2, 3, and 4. In recent year, my lab also started to work on the cap structure, not this M7G cap. In addition to this one, the first and second ribonucleotide on the ribose position, the 2' OH position, they can be methylated by 2 cap methyltransferase. So we also study this enzyme, and they have a lot of interest phenotype. I hope I can share with you next time. Okay, so back to the CPB protein. So this protein initially is identified by my mentor, Joe Ricker, in the oocytes, and later the Eric Kandel, as you all know, the Nobel laureate, worked at the learning and memory aplasia. And he also later on found this protein, the homolog in aplasia, is also important for learning and memory. So I guess initially with the CPB1, the function is well characterized in the mouse and in the aplasia. And in 2000, after the first human genome track is complete, and just by sequence search on the RNA binding domain, you can pull out so-called 2, 3, and 4. And although they share the sequence similarity with CPB1 only 45%, but among themselves it's very identical, and they can bind to the RNA in vitro with the same kind of the sequence. However, in vivo, through this year's study, we found they're quite different. Okay, and so initially we'll focus on 2, 3, and 4. It's also because there are new knowledge is known about it. Except the one study in the fly, OP2, showing the OP2, the fly homolog of the CPB 2, 3, and 4, is important for long-term Cauchy memory. And I think there's a study done by the Dr. Jiang Anshi's lab also show the OP1 in the fly is also important for memory. So indicate this protein has something to do with the memory. Okay, just for some non-neural science student here. So for the system, I'm working on the hippocampus. The majority of this is excitatory neuron kind of like this. Okay. So they have a lot of elaborate dendritic tree. This is where they're receiving the incoming information. And they will kind of compute and decide whether incorporate this information, whether they should trigger and continue to send this information to the next neuron. And this will be determined by the end of the axonal terminal, where there's a lot of synaptic vesicle inside has a neurotransmitter. And our system will be the amino acid glutamate, which is an important neurotransmitter in the hippocampus. And this release of neurotransmitter bind to the postsynaptic side and it's going to transfer the information again. So in reality, when we culture the neurons, and you can see this so-called dendrite region, there's a lot of protrusion, small protrusion structure. We call it dendritic spine. Okay, so when we see this kind of structure, we know they're postsynaptic in the dendritic region. And we actually do experiment to quantify the number or the morphology of the spine, because you can also indicate what's the situation about the communication between the two neurons. So why the synapse is so important and why we want to study it is simply because, as you all know, neurons are postmitotic cells. They don't really proliferate. We have very limited neurogenesis in adult. So the way really to support the tremendous computation and storage power of our brain or for your daily study is through changing the synapses. Okay. So simply speaking, this is so-called a pre and post synapse. When the postsynaptic synapse receives stimulation from the presynaptic terminal, they will undergo some kind of change. If this change exists when the next stimulation coming in, they are able to respond differently than the first time. So this kind of phenomena we call the synaptic plasticity. And we now also know for this change to last for short term and important for short-term memory, likely through like post-translational modification of the protein. However, for this change to persist for a long time, you require new protein synthesis to support either the structure or functionality of the synapses. And actually, this is what, more than 60 years ago, we already know long-term memory formation require new protein synthesis. And that's because of pyrimycin you might use in the lab, which is a protein synthesis inhibitor. And Dr. Fleischmann, actually, they train the mouse to navigate in the maze. And right after training, if they give it the pyrimycin and test the mouse again in 30 minutes, for example, and definitely the mice has learned the task can perform much better than the one without. However, if you inject pyrimycin, put it overnight, and next day when you come back, oh, this trained mice behaves just naive one. They don't remember what they have done before. So that's why people in the field are so eager to know what's all this protein component. If we know this protein component, can we know the molecular secret of the memory? Of course, that's kind of simplified thinking. But just to let you know how complex the system is, for mouse we work on, if you divide the synapse per each neuron, there's thousands of synapses in each neuron. And in human could be even higher, thousands or tens of thousands. Maybe smart people is even higher. Okay, so now the question becomes, if you want to modify this synapse for long-term memory, and where the protein comes from. So one way, of course, is from cell body. And there's a study showing in response to neural activity, there's some transcription can activate, and this RNA can be sent out to the synapse, or the protein can be synthesized, sent out to a synapse, and somehow be captured by activate synapse. And the other way, more precise and maybe more rapid response, is by through regulation of the translation locally in the dendrite, right? So if you have a dendritic localized RNA, you have a translational machinery there, they just off. But in response to stimulation, you turn on this machinery and you can quickly synthesize protein, and also in the region where you want, where the synapse has been stimulated. So I think that's the idea initially. We want to work on local translation or translational control in the neurons from the frog oocyte lab. So one way to support there's a local translation is because a lot of EM study demonstrate, this is just composite electron microscope data, and showing there's a lot of polyribosome and just sit beneath the synapse in the dendrite or in the dendritic spine. So indicate the possibility of a local translation. But when it comes to the experiment, different experts will use different approach. For electrophysiology, they do the electro recording. Here, just to show you, this is a typically hippocampus slice. They have a 2 layers of the neurons. And the information, the activation, this pathway from CA3 to CA1 neuron is very important for spatial memory. And here show you just the cell body, the dendritic layer, but they didn't show you the axon. So basically what we know is the CA3 neuron, they will project the axon into the CA1 neurons. And this we specifically called the Sheffer collateral pathway. So this pathway we record quite often in the lab. And so in the study done in Schumann's lab, they found using a BDNF, it's a neurotrophic factor that can induce the LTP. And this LTP, when you add a cyclohexamide, will be abolished. Indicate this LTP is protein synthesis dependent. So then they did a pioneer cut. They're the first one to cut. They cut the cell body layer from the dendritic layer and repeat this experiment again. They can induce LTP, and it's sensitive to cyclohexamide. So this conclude, at least for some form of this electrophysiological long-term synaptic plasticity can be supported by local translation. And here I want to emphasize, because it will be my today's focus of the talk, is that when we did this experiment, a lot of time just to avoid the complication. When you stimulate this axon, you don't want this electric signal also back to CA3 neuron and also project to other neurons. So we definitely will transect. We sect the axon from the cell body for doing the recording. But no one ever mentioned about why it's not due to the local translation in axon, why is it due to a local translation in dendrite? And there's some historical reason for that. Okay, I will mention that later. So the other way you can also study the local translation is by either, this is a kind of fancy image, you can have the GFT label mice, and now we have a way to localize specific dendrite, and you can monitor in vivo before and after 1 month. And you can definitely tell you, one can definitely tell you some of this kind of protruding thin synapse, they're not very stable, and they will be gone after a month. And some, initially it's not quite stable, they will become more mushroom-like spine, more stabilized. So this is in vivo data show you the spine are very dynamic. And we also can do biochemical isolation to pinch of this structure. This pinch of structure can be resealed as so-called a presynaptic vesicle attached to a postsynaptic compartment. And you can definitely tell this is presynaptic because they have a lot of bubble inside. And this is synaptic vesicle, inside has a neurotransmitter. And this is a postsynaptic density, so-called the density region is because they're highly electron dense. And the reason for that is just the postsynaptic side, this is just part of it. There's a lot of protein, thousands of protein clustered in here, able to receive this synaptic transmission signal and propagate to the downstream. Okay, so initially when people study on local translation or translational control, it's often in the early development, like in embryo. They found a lot of cases your protein or translation machinery can be localized in posterior and anterior of the embryo, and which will generate protein, create a gradient, and that's important for morphogenesis of the embryo. But in neurons, although they're not embryo, but you can imagine that the cell I just showed you, they have very elaborate structure. They have this dendritic structure, they have an axon structure, and the dendritic structure, at least in culture, can reach like 300, 400 micrometers that long for dendrite. For axon, we never quantify, I don't know how long it can be, but it can be pretty long. And some of the extreme case in the human, even some of the axon can